Good evening, guys. Uh, so, in this lecture, we'll be starting with the discussion that was taken place in the week 7 of experimental biology, sir, experimental biotechnology class. And in this lecture, we'll be discussing about various immunological techniques that are used to study various things in our cell biology experiments. Okay. So, starting with the first question, which of the following is involved in humoral mediated immunity? The options are T cells, B cells, antibody, or plasma B cells. What can be the answer? Okay, any more answers? Okay, so like the immunity that we are talking about in case of humans is divided into mainly two parts. First is the humoral immunity, and the second is cell mediated immunity. Okay, and this immunity we are talking about is called as adaptive immunity. Okay, so now in case of humoral immunity that we are talking about, we mostly talk about the proteins that are involved in the blood. Okay, so that is basically the antibodies, and the immune response is related to the antibodies. And in case of cell mediated immunity, we talk about mostly the T cells. Okay, and the immune response that is associated with the T cells and its following function. Okay, so in case of humoral immunity, we mainly talk about the antibodies, not the cells associated with it. Okay, so the correct answer is option number three, the B cells and plasma B cells. Okay, so just a brief about that. So B cells are the cells that arouse from the bone marrow, and those cells divide into two types of cells. One is the plasma cells or plasma B cells, and the other one is the memory B cells. No, I'll come to that. Okay, so plasma B cells basically produce the antibody. Okay, and memory B cells are like a memory of that particular antigen or pathogen that have been affected, and on that response these antibodies are being produced. So these memory B cells are stored in bone marrow. So when the second infection of the same pathogen comes, then these memory B cells become active, and they convert into plasma cells and the memory cells again, and then these plasma cells produce antibodies related to the same pathogen. Okay, now coming to the question that was in the chat. So, like, based upon the first interaction of the pathogen with the human body, okay, the immunity is divided into two parts. First is the innate immunity, and second is the adaptive immunity. Okay, so innate immunity is the immunity that is naturally present in our body, and it's the first line of defense of a pathogen. Okay, so for example, we have skin. Okay, different components of skin we have that basically helps us to protect from the outside environment. Okay, maybe the pathogens or the viruses, different cells are there that produce sebum. Okay, that also protects us from the different pathogens. Then we have different, then we have tears, the acid in the stomach. Okay, and different cells associated with it. Okay, so this comes under the innate immunity. That is a natural life of natural defense mechanism of our body. And secondly, this is non-specific. Okay, so that means even if the pathogen that is coming, for example, there is one pathogen, this is a bacteria, and there is another bacteria, okay, that comes to the body, and both of them will be treated in the same way as all of the organism. Okay, so this is non-specific in nature. And now we are talking about the adaptive immune response. So in this case, what happens, this is the thing that is adapted by our immune by our body during our life course. So for example, we are born, and then we move to adulthood. So during that point, when we encounter different pathogens, different environmental conditions, so on that basis our immune response become active and become responsive in that respect. Okay, so this is adaptive immunity. And then in adaptive immunity is specific in nature. Okay, so that means if, for example, we have bacteria 1 and bacteria 2, so different response will be there for bacteria 1 and different for bacteria 2. These will not be the same. Okay, and in adaptive immune response or adaptive immunity, we have two kinds, that is humoral or cell mediated. So humoral means with the help of the antibodies that is present in the blood, and cell mediated means with the help of different cells, mainly the T cells, okay, that is also present in the blood itself. So this is the case. So antibodies or humoral immunity do not come under the innate immunity, because this is specific for specific pathogens. Okay, so any more doubts in this question? So if not, then we'll move on to the next question. The Y-shaped structure of an antibody is composed of how many protein chains? What can be the answer? Okay, any more answers? Yes. So just a brief, we draw an antibody structure like this. Okay, so this is the light chain, that is light chain number 1, heavy chain number 1, heavy chain number 2, and light chain number 2. So there are two heavy chains and two light chains. So total four chains in the antibody. Okay, so the correct answer is option number 4. If the question would be how many types of protein chain, then it would be 2, because there are two heavy chains and two light chains. Okay, so yes, then let's move on to the next question. In which phase of the antibody production there is entry of pathogen in the body and contact with the immune cells? The plateau, log, lag, or decline phase? What can be the answer? Okay, any more answers? So what happens in case of antibody production? So the correct answer is option number 3, and here is the explanation. Okay. So whenever we have to produce an antibody or the body produces an antibody, so the first phase is, so this is like a general growth curve of of an organism. Okay. So first what happens in the lag phase, there is an entry of the pathogen. So this becomes the stimuli for production of the antibody. Okay. So now in the lag phase, the pathogen enters the body and it develops a contact with the immune cells. Okay. So now when the immune cells recognize the pathogen, they start expanding their cells. Okay, so this lag phase determines the interaction of the pathogen with the immune cells. Okay, now when they recognize that there is some pathogen inside the body, then they start expanding their cells, and the B cells or the plasma cells specifically, through clonal selection and all those theories. And then while expanding these cells, now they start producing the antibodies in the log phase. Okay, so they enter the log phase where they expand their cells and multiply into multiple cells, multiple plasma cells. Now they start producing the antibodies. Okay, so these antibodies titer increases significantly or exponentially in the log phase. Okay, now there is a plateau phase, that means the stationary phase. So now when the body recognizes that, okay, now the pathogen amount has gone down in the body, so now we have to limit our production of the antibody. So now what happens? The production of the antibodies is limited with the degradation. So now what the case was, the antibody production is also high, and at the same time the antibodies that have already been used for an immune response goes into the catabolism. Okay, so they are already degraded. So this means there is an equilibrium between the production of the antibodies and the degradation of the antibody. So this remains the stationary phase. Now, when the pathogen is fully removed from the body, then there is a decline phase that leads to decrease in the antibody concentration inside the body. So this marks the production of antibody and how it happens inside our body. Okay. So anybody has any doubt in this particular question? So titer means this is a different word for antibody concentration. Okay. So that is measured in the form of titer in case of antibody. So this is basically a concentration of the antibody that is present in our cell, in our body. Okay. If not, then we'll move on to the next question. So which of the following is an antigen presenting cells? So macrophages, T cells, activated B cells, or plasma B cells? What can be the correct answer? Okay. Any other? Hmm. So yes, the correct answer is option number one, the antigen presenting cells. And how basically antigen presenting cells that we are talking about are phagocytes. Okay, so these phagocytes take up the pathogen inside them, engulf them, and then degrade those pathogens and present some of the proteins of those pathogens onto their surface. Okay, for example, This is an example of a vaccine. Okay, how a vaccine works inside our body. So this is an antigen presenting cell. It engulfs this particular vaccine or a pathogen. Okay, now what happens? It degrades it through a degradation machinery that it has, because these are already phagocytes, so they have a efficient degradation machinery because of the lysosome or the proteasomal degradation machinery. Now, after degradation, they basically present these of the some of the parts of those pathogen onto their surface. And for example, here we are talking about MHC class 1. Okay, that so MHC is the uh, there are two kinds of receptors, that are MHC1 and MHC2. Okay, so these are histocompatibility complexes basically. Okay, so the what happens, the MHC1 presents the antigen to CD8 positive cells, CD8 positive T cells. Okay, so this becomes the cytotoxic T cells, and MHC2 presents it to CD4 positive T cells, that are T helper cells. Okay, so if we have to like present or initiate a cytotoxic T cell response, then MHC1 will come into picture and it will present it antigen to the TC cells or the CD8 positive cells. Okay, and if we are if they have to produce the antibodies based specifically, so APC will present the antigen with the help of the MHC type 2 to the CD4 positive T cells, that are the T helper cells. Okay, now T helper cells can now initiate antibody production uh, by activating the B cells. Okay, now B cells can produce the antibodies using plasma cells, and it will produce the memory B cells. Okay, now to remember how the which kind of MHC will bind to which receptor, so there is a trick. Okay, so as you can see that 8 is more than 4. Okay, so there are two students. One got 8 marks, so it went first, and the other one got 4 marks, that is, so it came second. Okay, so this was the trick that we used to use to remember which MHC bind to which kind of cells. Okay, so these are the antigen presenting cells. So if anybody has any doubt, can ask. So other examples of antigen presenting cells are B cells. Okay, B cells also sometimes engulf the pathogens and present on different cells. And the other ones are dendritic cells. So these are the examples of the antigen presenting cells. Okay, so if no doubts, then we'll move on to the next part. So now we'll be discussing about the hybridoma technology. Okay, so this process is used for the monoclonal antibody generation. So first I'll brief about the whole process, that how it takes place. Okay, so hybridoma technology is basically used for the monoclonal antibody synthesis. So antibodies are basically of two types: polyclonal and monoclonal. Okay, So in case of polyclonal antibodies, what happens? For example, this is an antigen and we have different epitopes of this particular antigens present on its surface. Okay, so epitopes are the sites that are present on an antigen and that is being bound by the antibody or recognized by the antibody. Okay, so one antigen can have multiple epitopes. Okay, for example, here I have shown you different shapes. Okay, now if in a population we have antibody for example, this is the first epitope, we have antibody against this particular epitope and this particular epitope and this particular epitope. Okay, so we have a mixture of all the antibodies against all of these epitopes. That is, that mixture of antibodies will be called as a polyclonal antibody, because the antibodies that are present in this particular solution is a mixture of antibodies against the epitopes present on this antigen. The antigen is same, but the antibodies are against different epitopes of the same antigen. Okay, now if there is a case we want antibody against this epitope 1 only, okay, and we get a pure antibody against epitope 1, then that is called as a monoclonal antibody, because that is only against the epitope 1 of this particular antigen. Okay, so am I clear with the difference between the polyclonal and monoclonal antibodies? Okay, now how do we get a preparation of this monoclonal antibody? So first what we do is we take a mice and we inject those particular antigen on the in the mice so that we get sufficient B cells. So basically we have to isolate the plasma B cells, because we have to take the, because these are the antigen antibody producing cells. Okay, they will produce the antibody. Now, those cells that we are isolating will be HGPRT positive. Okay, so what is HGPRT? This is hypoxanthine, uh, hypoxanthine transferase. Okay, I'll let you know the full form. So, what happens? It is involved in the salvage pathway. Okay, so now, first I'll explain what is the salvage pathway, just a brief. So there are two types of pathways that are depends upon the nucleotide synthesis. Okay, so one is the salvage pathway and other is the de novo pathway. So in case of salvage pathway, what we have the original bases that are triphosphates in nature, and using those bases, uh, we degrade them and form new bases out of those that can be used inside our body. Okay, so this is the preformed bases, then these are recovered to form the new bases. In case of de novo pathway, what happens? The bases, the nitrogenous bases basically, or the triphosphates that are used, no, salvage and de novo, both pathways are found in plants and animals. Okay, so in de novo pathway, we have different components that make these bases. So for example, we have different amino acids, different sugars, for example ribose, and one nucleotide bases like formate, vitamins, folate, and other things. So now these components make different bases. So this is the de novo pathway, making the bases from scratch basically. Okay, so now there is one component in case of salvage pathway, that is HGPRT enzyme. Okay, so this HGPRT is hypoxanthine guanine phosphoribosyl transferase. Okay, so it transfers the hypoxanthine residue to a phosphoribosyl, that is PRPP, and hypoxanthine is the precursor for guanine basically. Okay, so it's a precursor for guanine and it is first transferred to the PRPP precursor and then it is converted to guanine. The whole base is converted to guanine. Okay, so it is a main enzyme that is involved in the salvage pathway of the purines. Okay, and in case of de novo pathway, we have one important enzyme that is dihydrofolate dehydrogenase. So that helps basically formation of the different deoxy subunits of these particular bases. Okay, so that's why both the enzymes are important in case of the nucleotide synthesis. Now, moving back to the why we need these in our discussion of the hybridoma technology. So, what will we do is we will use other type of cells that are myeloma cells. Okay, why are we using myeloma cells in first place? Because B cells that we are using have limited time span. Okay, so because of their limited time span, they will not be able to produce that much amount of antibodies that we require for commercialization. Okay, so and these myeloma cells have HGBRT negative, so they do not produce HGBRT. That means they are solely dependent upon the de novo pathway for their nucleotide metabolism. Okay. And secondly, they do not produce immunoglobulin. That means antibody production is not there in myeloma cells and they are immortal. So we are utilizing the part immortal for the myeloma cell. Okay. So these B cells are antibody producing, but they have limited time span. But myeloma cells are not producing antibodies, but they are immortal. So we are using them for increasing the time span of our antibody production. Okay, so this was the first step in hybridoma technology. We chose the cells from the mice that are B cells, have HGPRT, and are producing the antibody that we need. Then comes the myeloma cells that are HGPRT negative, do not produce antibody, and those are immortal. Now, we fuse both of these cells in a polyethylene glycol. So, polyethylene glycol is a substance that is used to fuse both these cells without any. So, they fuse the cytoplasm, and then after fusing the cytoplasm, the nucleus all afterwards fuses itself. Okay, so after fusing this particular cells, we will get five populations. Okay, one would be unfused B cell, that means only single B cell would be there. Other would be unfused myeloma cell. Then there could be a possibility that two B cells have been fused with each other, and there could be a possibility that two myeloma cells have been fused with each other. Okay, now the last possibility and the ones that we actually need for our monoclonal antibody production are the heterokaryons or the fusion between B cell and the myeloma cells. Okay, so these fused cells we need actually for production of the uh, antibodies, monoclonal antibodies. Okay, so till here, is there any doubt or am I going too fast? So till here we have got five population of cells. First is the unfused B cell, the unfused myeloma cell, B cell fused with the B cell, myeloma cell fused with the myeloma cell. And the last population is a heterokaryon, that is B cell fused with the myeloma cell. Okay. Now we separate out these cells, and because we want the heterokaryons between B cell and a myeloma cell. Okay. So we use a hat selection medium. So what is hat? So hat selection means HGPRT, oh sorry, hypoxanthine, guanine, hypoxanthine, aminopterin, and thymine or thymidylate basically. So what is the role of all these three? Okay. In case of hypoxanthine, it's a substrate for HGPRT. Okay, so when HGPRT would be there, it would help, hypoxanthine would help to activate HGPRT. So it's basically a food. Okay, now myeloma cells do not have HGPRT, so they will not require hypoxanthine at all. This is for the B cells. Okay, so this is for the B cells only. Now coming for the aminopterin. So aminopterin is a inhibitor for dihydrofolate dehydrogenase. Okay, so dihydrofolate dehydrogenase is an important enzyme that is involved in the conversion of ribonucleotide to deoxyribonucleotide. Okay, so this dihydrofolate dehydrogenase is inhibited by aminopterin. So now what its role here? Now, in the myeloma cells, we are deficient of HGPRT. Okay, so that means salvage pathway is not operating in the myeloma cells. Now we are left with only the de novo pathway. After using aminopterin, the dihydrofolate dehydrogenase is inhibited. So in that case, what happens? This conversion cannot take place. So this stops the de novo pathway as well. Okay, so if a cell needs to survive in a medium, then it would require either of these two pathways to be functioning. But here, the cells do not have the salvage pathway in a genetic condition, and we are inhibiting the de novo pathway as well. Okay, so after growing these cells in a hat selection medium, the myeloma cells that are either unfused or the two myeloma cell fused will die out because of the use of aminopterin. Okay, and how will we now, out of the five population that we have, so we have B cell, myeloma cell, BB fused, MM fused, and the BM. Okay, so out of these population, myeloma cells are wiped out because they cannot grow without the nucleotide synthesis. Now, how will we separate the B and the BB fused? So, as we talked about, they have a very limited time span. So, they will die out eventually. They will not grow in the medium. Okay, and there is only a specific medium that will be allowed to grow the B cells. So we will not culture out these, and we will subculture all the cells that we have various times, maybe 5 to 6 times we subculture, so that we get a pure population of the B cell fused with the myeloma cells. Okay, so now these hybridomas. Okay, so now these heterokaryons will be called as the hybridomas. These are HGPRT positive because of the B cell. They are Ig positive, that means they produce the antibodies, and they are immortal. Okay, so we can subculture them and grow in a petri dish inside the lab conditions. Yeah, that's what I was talking about. So in this case we'll subculture, and eventually during subculturing the B cells or the fused B cells are wiped out, and we remain with the BM cells only. Okay, and these BM cells we'll subculture inside the medium, and then what we'll do, we have to generate the antibodies, the desired antibodies. So then what we do is we either expand them in vitro, that is in culture, and we isolate the monoclonal antibodies, or we generally like a virus, we inject it in mice, and we collect the ascites fluid that is developed because of the uh, growth of the tumor. Okay, and then from that we isolate the monoclonal antibody. So this was the whole procedure of how the hybridoma technology takes place for the production of the monoclonal antibody. So any doubts? Okay, you can ask afterwards also. Let's move on. So based on these, I have like developed some of the questions. So how are the monoclonal antibodies produced? Here are the options with you. Please select the correct option. Okay. Any more answers? Yes, the correct answer is option number two. So, as we have discussed just now, we have collected a single B cell clone that produces an antibody for the single epitope and fusing it with the myeloma cells. Okay, so let's move on to the next question. Hat medium inhibits. Yes, it inhibits the nucleotide synthesis. Okay, so I hope now you guys have understood the hybridoma technology and the uh, basic concept behind it. Okay, okay. So let's move on to the next question. Which of the following is a carrier used as a carrier particle in indirect hemagglutination reaction? The RBCs, WBCs, bacterial cells, or platelets? Okay. So first of all, the correct answer is option number one, that is RBC, and here is the explanation. Okay. So agglutination, from agglutination we means that clumping, okay, or basically when the membrane of RBCs broke off, then it releases all the components that are present inside the RBC. So that means that it has been clumped. Okay, so this is the real meaning of the agglutination that we are talking about. So what happens whenever we are resuspending RBCs in PBS, and so they settle down in a pellet form. Okay. Now, when we have to isolate some of the viruses, or we have to check whether any virus infection is there inside our body, then what we do is we resuspend our RBCs with the viruses, and then we pellet them down, and there is no significant pellet observed in that case. So that means what happens? The virus gets attached to the RBC surface and it basically lies the RBC. Okay, so this is called as clumping of RBC. And how we are using it for indirect hemagglutination? So in that case, if we have to check for any soluble antigen in our blood, okay, so there are various soluble antigens that are present in our blood. So we have, if we have to isolate them or we have to study them, then what we do is we use RBC as a carrier. They will absorb the soluble antigen on their surface, and then we recognize them on the basis of the. So clotting and clumping are two different things. Okay, clotting means there is a mass of platelet that has been formed. Okay, this is a mesh of platelets that have been formed, and in this mesh there are different proteins, clotting factors, platelets that basically helps a wound to be filled up. Okay, so in this case the major factors are platelets and clotting factors. Okay, now if we talk about clumping, so in that case, clumping means basically RBC lysis in simple terms. Okay, so RBC lysis can happen when there is any pathogen or anything that are degrading our RBCs. So that is called as clumping. So this is the basic difference between clotting and clumping. So this is basically in vitro assay. Okay, so we have to isolate the blood from an organism, and then we are using it to resuspend our like we are using it for our study so that we can test the soluble antigens that are present in our body. So this all the things will be done in vitro only. We cannot do anything inside any organism. Okay, so any more doubts in this particular question? So if not, then we'll move on to the next question. So which of the following diseases can be diagnosed using the indirect hemagglutination reaction? Okay, so someone said influenza. So generally, indirect hemagglutination is used to study viral diseases. Okay, because viral particles are generally soluble inside our blood. Okay, so these are soluble in nature. So that's why we need a carrier to detect these viruses inside our blood. So that is why indirect hemagglutination is done for viral diseases mostly. Okay. So in out of these, the viral diseases is only influenza. Okay. So the correct answer is option number one. Malaria is caused by a protozoa. Okay. So let's move on to the next question. Now we are moving into the precipitation reactions. Okay, so the Outerloni double immunodiffusion technique is used to, the options are here: determine blood type, detect specific antigens in a sample, quantify antibody concentration, or purify the antigens. Someone said second. Any more answers? Yes, the correct answer is option number 2. And here is the explanation. Sorry for the quality of the image, but so what happens? What we generally do is we make a agarose gel uh, base, and from that base we take out some pores using various needles. Now, out of these pores, in one of the pores we add the antibody or the anti-serum that is isolated from our own body, and in the next two wells we add the specific antigens. Now, both, because agarose is a porous substrate, so as we have discussed in the electrophoresis, so it has different pores. Now, what will happen? Both antigen and antibody will flow towards each other. Okay, and there will be a site where the interaction between the antigen and the antibody happens. Okay, so that will be seen as a white line. As you can see here, these white lines are basically the interaction between the antigen and the antibody. Okay, so now what happens, that if there are different like patterns that we can see, that how we can detect the antibody and the antigen. So these are the different patterns that can be seen, and this means that if, for example, like this uh, lines are seen, so this means there is uniform uh, yeah, there is uniform specificity between the antigen and the antibody. Okay, that is why it's straight line. Now, if we can see like these lines, so these means there is partial identity between the antigens also. Okay, and this means that it's full identity between the antigens, so antigens also can interact with them. Okay, so this is how we detect specific antigens from different antisera, or if we have to detect any antigen from, for example, any pathogen that is present inside this well. So we use Octerlony double immunodiffusion method. Okay, so any doubts in this particular part? So this is basically cross specificity between the antigens also. Okay, so that means both of them, the antibody that we are using has specificity for both of the antigens. So this is cross reactivity in short. Okay. Okay, so let's move on to the next question. Which parameter can be estimated by measuring the diameter of the precipitin ring in the radial immunodiffusion method? What can be the correct answer? So the correct answer is option number 4, that is the antigen concentration. Okay, so now how do we do the actual method? Okay, so what we have is we have a plate like this, as you can see in the image. and then we develop certain wells inside this agarose plate itself. And in this agarose, we have already mixed with a antibody of our interest. Okay, that will help us to identify that particular antigen present inside the sample that we have. Okay, so while preparing this agarose, what we do, we add the agarose to our buffer and then we heat it. Okay, after heating it, we allow it to cool down to such an extent that it does not freeze. Then we add the antibody, because antibody is temperature sensitive. So whenever we add the antibody, the temperature of the agarose must be such that the antibody is not degraded. Now, when we have to form a substrate, so we add into a petri plate. Okay, so after adding this mixture into a petri plate, it will appear like this, and we then develop some wells. Okay, as you can see here. Now, in these wells, we add different concentration of the antigens. So, this radial immunodiffusion can act as two ways, as a qualitative or as a quantitative. Okay, so in case of qualitative, we'll be able to detect the antigen on the basis of the ring. If the ring is formed, that means the antigen is present. Now, if we have to turn it into a quantitative method, then we will have to add different concentration of the antigens inside the different wells. Okay, so this is the increasing concentration of the antigens, and based upon that, the ring will be formed. Because if there is more concentration of the antigen, it will flow to more like towards more it will form a larger ring. Okay, and this ring is formed by the precipitation of the antigen with the antibody, and this ring is called as the precipitin ring. Okay, so now as this ring grows bigger, that means it is the concentration of the antigen is more. Okay, so now based upon this, we can form a graph, and based upon the diameter of the precipitin ring, we can estimate the antigen concentration inside that particular way. Okay, so for this, we have to form a standard curve, and based upon that, we'll be able to detect the antigen concentration of the unknown sample. So this is how the radial immunodiffusion assay takes place. So any doubts? So because this is a precipitation reaction. Okay, so now the precipitation or the molecule formed after the reaction of this antigen and antibody, that is called as a precipitate. So that is why this ring is formed by the precipitation reaction between the antigen or antibody, that is a precipitate of antigen or antibody. That is why it is called as a precipitin ring. Okay, so if no doubts, then we'll move on to the next question. The main molecule used in a pull down assay is typically a small molecule drug, an antibody, protein of interest, or DNA plasmid. What can be the correct answer? Okay, any more answers? So the correct answer is protein of our interest. Okay, and here is the explanation. Okay, So for example, if we have to study protein protein interaction, okay, so there is a signaling molecule that interacts with multiple partners inside a cell. So what we do is we isolate the cell lysate from our cell, and that is coded with the particular protein that can be GST or any other major molecule or an antibody. Okay, so antibody must have a site for this particular protein. Now what we do is we have a bait. Okay, so we know that this bait interacts with many proteins inside the cell. Okay, so this acts as a probe. So probe is used for identification basically. So this probe bait, when added with the cell lysate, it interacts with different proteins inside the cell lysate. Okay, so now this gives us an idea. Okay, this bait is interacting with different proteins that might be of our interest. So to isolate these particular proteins that we have to study, we use a bait that interacts with that particular protein, and then we isolate that particular protein and identify it. So, in case of a pull down assay, we have a bait molecule that is a probe that identifies the molecule or the protein that is of our interest, and that is used to basically study different processes inside the cell. So, am I clear? So, GST is glutathione transferase, basically. Here we can directly use the antibody also for detection of those particular proteins interacting with another protein. So, if no doubts, then we'll move on to the next question. The target molecule or the antigen in a sandwich ELISA is captured between which two antibodies? What can be the correct answer? Okay, so we got one answer, that is third. So yes, the correct answer is option 3, that is the capture antibody and the detection antibody. And here is the explanation. So in case of a sandwich ELISA, we have a antibody that is attached to a support or the substrate, and then we add the antigen of our interest, that is the serum or the blood, that will have the antigen that needs to be detected. Okay, now for the detection of that particular antigen, we add a detection antibody that is linked to an enzyme. Okay, so this enzyme would be have a certain particular substrate that will give us some color or a light that we'll be able to detect whether our detection antibody is bound to the substrate or not. Okay, So in every ELISA, indirect, sandwich, or competitive, we would have two kinds of antibodies. One is the capture antibody that would have the direct connection with the antigen, and second is the detection antibody that would be allow us to increase the sensitivity of the reaction. Okay, so this would give us some kind of result that would allow to detect the particular antigen that is present in our sample or not. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. How does a competitive ELISA works? What can be the correct answer? Okay, so the correct answer is option number one. Okay, so here is the explanation. Okay, and in my opinion, both the one and two are correct. Okay, both one and two can be correct because what happens in a competitive ELISA basically, we have either we have a labeled antigen or a labeled antibody. Okay, so what happens in case of a competitive ELISA that is commonly used, we have to label well with a known antigen that we have to detect. Okay, now what we do is we add a and add our samples that would have the antibody against this particular antigens. So we add our samples. Okay, so this would have the antibody of our interest that would have bind to these particular antigens. Now here comes the second part, and that is the competitive step. Okay, so now what we do is either we use a labeled antigen or we use a labeled antibody. Now if we use a labeled antigen, that means the antibodies that are free in this particular environment that would be able to bind to this particular antigen will be detected, and this antigen then we'll use for either color generation or detection basically. Okay, now in the second step, if we are using labeled antibody, so in that case, what will happen if there are any of these antigens that are remaining? Okay, now these antibodies that are labeled will bind to these particular antigens and will detect the signal. So now how the signal will be detected? So that means in first case, if the we are using labeled antigen, in that case the antibodies that would be free to bind. Okay, because most of the antibodies would bind to the antigen that we have. So the antibodies that would have been free to bind to this particular antigen will give the result. So in that case, the color or the detection that we are doing would be less because most of the antibodies would have bound to this. Okay, so and in the second case, if we are using labeled antibodies, okay, so that would detect in indirect way. How? If we are using the sample that would have the antibodies more in number, that means the antibody that is less labeled that will be binding to the substrate in less amount. So it will give less color or less detection. Okay, and if the more number of spaces would be free, that means our sample would have less amount of the antibody of our interest, and then it will be giving more detection, more color. Okay, so it will be basically inverse relation between the two. Okay, so this is how a competitive ELISA works. So any doubts in this question? Okay, if not, then we'll move on to the next question. The purpose of membrane blocking in Western blotting is, what can be the correct answer? Okay, any more answers? Okay, so the correct answer is option number three. So remember, when we are using any detection method utilizing the antibodies, so in that case, what we do is we block the surface first. There are different methods. People use detergents, people use BSA, FBS. Okay, all those proteins are used for blocking. So in that case, what happens if, for example, this is the protein that we have to identify, but there are other sites in the region also that can bind to the antibody also. So first we have to block these surfaces so that there is no non-specific binding of the antibodies in this region, and the antibody bind to this particular surface only. Okay, so this would, what would this do? This would increase the specificity and the signal of those particular proteins, and secondly reduce noise. Okay, so this would be the correct answer. So to reduce noise and the non-specific binding of the antibody. So in any of the assays utilizing antibodies, this blocking step would be necessary. Okay, so let's move on to the next question. The reagent used to develop an HRP based secondary antibody system in Western blot is, okay. So HRP means horseradish peroxidase. Okay. So as it is a peroxidase, so H2O2 and luminol would be the correct answer, as it acts as a substrate. Okay. So let's move on to the next question. Queen 20 is used in Western blotting to ensure proper blotting of the primary antibody, facilitate the transfer of proteins onto the nitrocellulose, ensure proper binding of secondary antibody, or reduce non-specific binding and protein-protein interaction. What can be the correct answer? So yes, the correct answer is option number 4. So queen 20 is basically used for blocking. Okay, there are different antibodies that are compatible with queen 20, and some like we use uh, that is Titan X100. So that is also a detergent. Some antibodies are compatible with Titan X100. Okay, so let's move on to the next question. The antigen antibody interactions involves covalent interaction between the epitope and the Fc region. Yeah, it was luminol and hydrogen peroxide. So antigen antibody interaction involves option 1, covalent interaction between epitope and Fc region, non-covalent interaction between the epitope and the FAB region, non-covalent interaction between the antigenic determinants and the Fc region, or irreversible covalent interaction between the epitope and the FAB region. Okay, any more answers? So inside our body, any interaction that is taking place, all the non-covalent interaction will take into place. Okay, being it enzyme substrate or antigen antibody. This will not be a strong interaction. Okay, these can be broken off inside the body itself. Okay, so the correct answer will be option number 2. And these are the interactions. Okay, just a second. So this is our antibody and this is a FAB region. So antibody have two main regions. First is the FC region, that basically is due to the crystallizable part. Okay, because when the people were studying different antibodies, so this constant region can easily be crystallized, so this was called as the FC region. And F fragment crystallized, and the second fragment that was used for binding to the antigen was FAB. So this is an antigen F binding fragment. Okay, so this FAB region is made up of one light chain, half of the light chain, and half of the heavy chain. And the constant region is only made up of the, sorry, FC region is only made up of the heavy chain. Okay, and this FAB region has a antigen binding site. Okay, and these antigen binding sites are basically involving different non-covalent interactions like salt bridge, hydrogen bonding, van der Waals forces, pi-pi interactions to bind to the particular epitope inside the binding site. Okay, so now these epitopes that we are talking about. Okay, so as I told before also, an antigen is a bigger protein. Okay, this antigen would have different parts that can be recognized by a antibody. So these parts are basically the epitopes that are recognized by different antibodies. Okay, so an antigen would have different epitopes, and different antibodies can recognize these particular epitopes. Okay, so an antibody will be able to bind only to a epitope of that particular antigen. So any doubts in this particular question? Okay, if not, then we'll move on to the next question. The parameter to define binding capacity of an antibody is specificity, cross reactivity, affinity, or avidity. what can be the correct answer. So, yes. So basically, if we are talking about an antibody, we have different uh, terms that are used. Okay. So, for example, I'm drawing one antibody. This is the heavy chain, the light chain. This is the antigen binding part. Now, if an antibody can bind to this particular epitope, that means it's specific for this particular epitope only. It will not be able to bind to this or this epitope. So, the first thing, specificity, is this part. It is specific for only one pathogen. So, this kind of antibody, for example, IgG or the different antibodies that we have, is specific to different epitopes. So, one antibody cannot, like, sometimes it can, if there is certain region of similarity between the two epitopes, then one antibody can bind to the other antigen, and but generally it is specific for a single epitope. Okay, so this is specificity. Now, as I said just now, that if there is similarity between two epitopes, that means one antibody can also bind to the other epitope also. Okay, so that is also called as cross reactivity. Okay, the third thing is affinity. That means how strongly the epitope is binding to this particular antibody. Okay, so for example, if we have a shape of the antigen binding site like this. Okay, so now this is the region where the epitope will bind. If the epitope shape is like this, and like this, so out of these three, this would have the higher affinity to bind to this as compared to this particular epitope and this particular epitope. Okay, so this means that this has higher affinity to bind to this epitope, and secondly, if it binds to higher affinity to this particular epitope, the antigen binding region, that means it will not be able to release this epitope easily. Okay, so the affinity terms is derived from the dissociation constant, basically in mathematical terms. Okay, so this was the affinity wala part. Now, how many, for example, if one antibody have two antigen binding sites, the number of antigens that can be bound to one antibody is two. Okay, so the fourth component that comes to an antibody is the number of antigens an antibody can bind. So this number is called as the avidity. Okay, so for example, this is an antibody that can bind to two antigens. So it can be the capacity of this antibody is two, and the avidity is also two. So that means the number of antigens or the that can be bound to this particular antibody is two. Okay, there are antibodies, different antibodies that can bind to more number of antigens also. So that means their avidity is more. Okay, so as per the question, the binding capacity of an antibody is defined on the basis of its avidity. So correct answer is option number 4. So you can ask if there is any doubt in this question. Okay, if not, then we'll move on to the next question. Which of the following techniques can be used to study protein-protein interaction of a transcription factor X. Fish or fluorescence in situ hybridization, SGS page, chromatin immunoprecipitation, or pull down assay. This would be pull down assay. What can be the correct answer? Okay. So what happens? These are the different techniques that we use. So first is fluorescence in situ hybridization. So here, what we do is we generally use it for detecting the nucleic acids. Okay, so we label the nucleic acid in such a way that it will be able to detect the particular or specific nucleic acids that we have. SGS page will not be able to detect the protein-protein interaction. It will only be telling us the size. Okay, there are different methods like EMSA or other methods that can be used, but SGS page like cannot be used because SGS and beta mercaptoethanol denatures everything. Okay, then we have chromatin immunoprecipitation. So this helps in detecting the protein and DNA. Okay, so when a protein binds to specific DNA site, that is able we'll be able to detect using chromatin immunoprecipitation. And the last one is the pull down assay that we discussed a few slides back. So in that case, what happens? We have a like here we use the GST tag with a probe. So this probe will allow us to check for different protein interactions in our set. Okay, so the correct answer is pull down assay. So any doubts in this particular question? Yes. Yes, that's what I explained. So, for example, if two proteins are interacting, okay, so how will they interact? For example, this is a protein X that has to give its signal or activate it by interacting with a protein Y. Okay, now this interaction is non-covalent in nature. Okay, that can be hydrogen bond, okay, or van der Waals forces, or salt bridge. Okay, no covalent interaction would take place here. Now, if we are using SDS, okay, so this would break all the interactions between both the proteins, and this will denature also the both the proteins by implying the negative charge. Okay, so if we have to study the protein-protein interaction, then we will not be able to study with using the SDS page because this interaction that we are talking about must be intact. Then only we'll be able to say, okay, X protein is interacting with Y. So using SGS page, we will not be able to detect the interaction. If we are using normal page, okay, the native page that we talked about, so in that case we are not using any denaturing agents at all. So how will we detect using page only? We have three wells, for example. In one well we'll add protein X, in one well we'll add only protein Y, and in the third well we'll add both protein X and Y. And if we can see, the two bands of protein X and Y are here, and the interacted protein that we talked about is here. That means these two proteins are interacting with each other. Okay, so is the doubt clear? Okay, so this marks the end of our today's lecture. So if you guys have any doubts in the lecture of either of these questions or from the lecture, you can ask. Okay, thank you. Okay, then we'll close the session for today. Thank you guys for joining. Yes. Uh, for this particular topic, uh, I'll suggest QB. Okay, so QB is a immunology book. So the immunological techniques are better explained in QB, and rest like for the other techniques that we are studying. So Wilson Walker is one of the books that I preferred for electrophoresis and chromatography, and the rest I'll let you know. Okay, so for immunological techniques, we can use QB. Okay, so this can be used for studying these techniques. Okay, so let's close this session for today. Thank you.